Okay, so there's a couple virtual labs for this that exist, and I really like them, but there's something about me that wants you guys that are at home to kind of see what we're doing exactly in class. So I think I'm going to record this. I tried to do this at the same time I was doing the experiment with the students in class yesterday, and it was a little bit difficult for me to manage both at the same time. So I'm going to go ahead and record this one because I had such fun doing it by myself last time too. Um, so what we're trying to do is this E. coli here that's growing does not have this plasmid in it. This is just grown on what's called LB medium, which is like a food for E. coli. And you can see that there's a lot of growth here and then a little bit less. And then this is the part that they have isolated colonies. So this bacteria does not glow in the dark. It doesn't have the plasmid that it needs to get to do that. So what we're going to try to do is put that plasmid into the E. coli. Now, we want to make sure that we're looking at just the colonies that have, oh, you're fine. Just the colonies that have the plasmid in them. So we're going to try to sort out these that don't have the plasmid for ones that do get the plasmid. And the best way to do that is to use antibiotic resistance. So what we're going to be doing in these plates is we're going to be putting them on plates that have the antibiotic ampicillin, which is very similar to penicillin, ampicillin. And that most bacteria will be killed by ampicillin. But because this plasmid has engineered into it an antibiotic resistance gene, if it gets the plasmid, it will be able to grow on ampicillin. And then the other thing that we want to do is we want to really produce this green fluorescent protein in large enough amounts that we can see it pretty easily and quickly. And what Biorad, who engineered this plasmid, pGLO, did is they put the green fluorescent protein gene, which you see here, under the control of inducible promoter. In this case, it's arabinose. So if arabinose is present in the media, it will, and ampicillin, it will produce colonies that are expressing the green fluorescent protein. If there is no arabinose, then it does not express green fluorescent protein, but since it got the plasmid, it will still be resistant to the antibiotic. So you'll see those results in here in a minute or so. Well, more than a minute, but get the idea. All right, so let me see if you should have read this already. And then I'm just going to get to it. So I'm just going to follow the instructions. You can follow along with me. And what it wants me to do is to get a pink and yellow tube and to put transformation into open the pink and yellow tubes and transfer using a sterile pipette. I might have to get some more of these. 250 microliters of the transformation solution, which is in the blue cup here, or the blue tube, 250 microliters. So I have to see on this, I think that's up to, I know it's right up to there. That's the 250. And I'm going to put that into each of the pink and yellow tubes as it is asking me to do. 250 microliters of transformation solution into each of those tubes. Okay, so there's two. Let me make sure that's 250. Yeah, that looks like 250 in the minus. $250 in the plus. And I think I'm done with that for now. And then the next thing it wants me to do is to close the tubes and stick them in crushed ice. Okay, I could do this. Put everything into this crushed ice. Okay. All right, now what it wants me to do is to use a sterile loop and pick up 2 to 4 colonies and put them into the minus tube first. So I'm going to go into this plate here, and I'm going to pick 4 of these colonies here, and with my sterile loop. So let's see if you can see those. There we go. And so I'm just going to try to pick up 4 of them. Trying to find the area that has a good 4 in it. Just going to go for those right there. Okay. And then those are going to go into the minus tube. And it wants me to put them in there and then do this. So they are going to be mixed up really well. And then I don't want to reuse this loop. I'm going to set that aside and put it into my biohazard. And then that's going to go back on ice. And then it wants me to do the same thing for the positive tubes. I'm just moving along here with our instructions. Get the same thing and pick up 4 colonies for the plus tube. So same thing here. I'm going to go in and pick up 4 colonies best I can. Let's see. Okay, it's not going to look like much, but they're there. And then I'm going to put that into my plus tube. And it's going to be mixed up really good, so I know that there's cells there. And then I'm done with that loop, so I need to get rid of it. And then it wants me to then take a micro pipette, which is this, and make sure it's on 10. And get my plasmid DNA, 10 microliters. That's in this little green tube here. It doesn't look like much, but 10 microliters is there. And to take that 10 microliters, I'm going to use my micropipette, suck up the 10 microliters. That tube is done, and I'm going to put it into the tube that says plus. Obviously, I'm not going to put it into the one that says minus. And I'm going to go right into the tube there and pipette that 10 microliters right into there, into that tube. Then I can get rid of that pipette tip. And I think I'm done with my micro pipette. And then these have to sit on ice for 10 minutes. So let me set a timer for 10 minutes. And that will incubate on ice for 10 minutes. Okay. Just want to make sure that the ice is there. Okay. Okay, so what we're going to do now is talk about these plates that we're going to grow these bacteria up on and what is contained on these for while we're waiting that 10 minutes. Okay, so what this material that this E. coli is growing on is basically food for the bacteria, and it's in a form of a broth called luria broth. It's almost like chicken broth in that it has a lot of different nutrients in it. And that broth, we can add a few different things in it to select the bacteria for what we want them to be able to have in them. Try to straighten this out a little bit. See, my cat scratches on me. Okay, so the very basic of this plates, this is got the food for the bacteria to grow on, and then agar. So it's easier to work with bacteria if they're on a solid or semi-solid surface. So that's why we use agar. And then some of the plates have, that one is just the food. This one is the food plus the antibiotic ampicillin. This one is the same, the food LB plus the ampicillin. And then finally, this is the one that is going to be LB plus the sugar arabinose and the antibiotic ampicillin. So as I said before, on this plasmid that we're going to put into the bacteria, it has an ampicillin resistant gene in it. And it also has an arabinose promoter that if the arabinose is present, it will express the GFP. So it's a lot like the lacoperon in that regard. And so this one here is LB, the food plus the sugar plus the antibiotic resistance. So what we're going to do here is we are going to label our plates accordingly to what they're going to get. So we're going to have one that is going to be, let me just line these up here. And so LB amp era, LB amp, LB. Okay. So this one is not going to get the plasmid. So I'm going to try to write these as much as I can on the edges so it doesn't take up my whole plate when I'm going to look at it. So this one is minus pGLO, LB, and I want to put my initials on it and the date. So there's going to be a lot of stuff on here. Today is the 11th. And I also want to put that this bacteria that's going on this is E. coli. Okay, so that's all I need to put on that plate. And then this one is going to be minus pGLO. And it's also going to get the same information. It's going to get my initials and it's going to get the date 3/11. And then it's going to get E. coli. All right, this one is going to be plus pGLO, KG for my initials, E. coli 3/21. Not 21, that's my mom's birthday, 3/11. And then this one is going to be plus pGLO, KG 3/11, E. coli. Try to label them on the, well, I'm just talking like I'm talking to people in the class, but try to label them on the side so you don't take up the majority of the plate in all writing. So let's talk about what should be found on these plates when they have to incubate for about 24 hours in order to see. So I'll take them out of the incubator tomorrow or Saturday. I'm not sure. Depends on when I can get up here. All right, so let's talk about this plate here first. This one is not going to get the plasmid, but this is just the food. So it doesn't have the antibiotic in it. So this really should grow everything that we put on it. It doesn't have the plasmid, but it also doesn't have antibiotic. So everything, we should get a lot of growth on this plate, right? This one here, this one is going to have the this one does have the antibiotic, but it's not going to get pGLO. So everything that would grow on this plate shouldn't grow on here because if it doesn't have the plasmid, then it doesn't have resistance to antibiotics. So the antibiotic would kill it. And so we're not expecting to see growth on this plate. This plate here, this one has the plasmid plus pGLO and ampicillin. So it should grow because it has the plasmid in it that has the ampicillin gene in it. But notice there's none of the sugar arabinose in here. So while we're expecting growth on this plate, we're not expecting the production of the GFP because that is going to be under the influence of the arabinose promoter. So in order to get that green fluorescent protein made, we have to have the plasmid, we have to have the arabinose, and therefore we should have about the same amount of growth on these two, but these colonies should glow in the dark because they have arabinose and the plasmid, and this one just has the plasmid with the antibiotic, so it will be not having the fluorescence. So that's what should happen. All right, so we'll get back to those in a minute. Let's see if there's anything else we need to do before this next step, which is going to be in 2 minutes. So it's actually making good real time here. So what we're going to do next is we're going to heat shock the bacteria for exactly 50 seconds. And it's not super hot. Your body temperature is 37. So 42 is not super hot, but we don't want to get it too hot and possibly kill the bacteria. So we're going to do it at 42 degrees Celsius for exactly 50 seconds. And then we're going to put them right immediately back on ice for a certain period of time here that we see. So I'm going to pause this for a second and I'll be back when we're done with the heat shock. So the heater is in a different part of the room. So I will come back after I put them on heat for 50 seconds. Okay, so those came back from heat shock. And now I am going to stop. I'm going to time for 2 minutes. And now what it wants me to do is to take this LB, which is bacteria food, Luria broth, and since we just heat shocked them, we're going to give them some nutrients to get them to kind of be a little bit happier. And it wants us to do 250 of LB in each tube. So I'm going to go ahead and take a new pipette and get 250 microliters of LB into my tubes. Kind of give them a little stir here. And then another 250 microliters into the +2, just to kind of get them to start in their growth. Okay. And then put those are going to sit at room temperature for 10 minutes. Oops. I'm going to set my timer for 10 minutes again. Stop recording. Okay, so this is a schematic drawing of what's happening here. And we use transformation solution, which is positively charged, and that's calcium, and it'll put like positive charges all over this DNA. And the membrane is negatively charged. So they're gonna, and the heat shocking kind of created like almost little holes or gaps, and then the plasmid can get sucked in that way. And then the plasmid, once it's in, it's going to make the gene for ampicillin resistance, or it's going to make the enzyme for ampicillin resistance. And also will, if arabinose is present, will produce the GFP protein that would glow in the dark. And you could see here on this part, it's just showing how in the, let's see here, in the presence of arabinose, you are going to get the expression of the GFP gene. If you don't have arabinose, then it doesn't transcribe and translate the GFP gene. One other thing to kind of point out here is when they clone this gene into the plasmid, they had to make sure that they only had the exons only, because it otherwise it wouldn't work being in bacteria. And I'm not going to worry about that retroviruses right now. All right. So we're ready to move on to this part. And I'm just going to go ahead and put my plates in that order. And so I need the plus pGLO, plus pGLO amp, plus pGLO, the arabinose, minus pGLO, ampicillin, and just the regular thing. So since this doesn't have pGLO, but it doesn't have antibiotic, we're expecting a lot of growth on there. So I'm just going to write like 4 pluses. And it's not going to glow in the dark because it doesn't have the plasmid. And then this one has the ampicillin resistance, but no plasmid. So we're expecting no growth on this one. This one is going to get the plasmid that'll give it the ampicillin resistance. So we should get a decent amount of growth. But since it doesn't have the sugar arabinose, we'll get the same amount of growth here, but this one should also glow because it has, oh wait, I messed these two up. This goes here. That one has the arabinose, so that should glow. And then this one is not going to glow. Sorry about that. I just misread which plates were which. All right, so let me see where I am for time. I still have 4 minutes. I'm going to go ahead and pause this. Okay, so I'm going to do my minuses first. And we should flick this to kind of mix it up, make sure that we have everything. Sometimes cells will, since they're bigger, they'll settle down to the bottom. So we just want to give this a little flick just to make sure that it's good. And it wants us to pipette 100 microliters into each plate. So 100, that's 250, 100, that's right there. 250, 100 microliters. Just make sure I don't have a bunch of bubbles. Sorry about that. So right there is 100. And that's going to go into the middle here. And then I'm going to take another 100 microliters and put that onto the ampicillin plate. And I'm done with that pipette. And while I'm waiting for that, I still have a timer going on the pluses. What I'm going to do is take a separate pipette or not a pipette, a separate sterile inoculating loop and spread out that bacteria across the whole plate. So it's evenly spread out, and try not to, oops. Okay. And then do the same with the second one. Just go ahead here and spread that out so it's not just stuck in the middle. Okay. Okay. Oops. Now these are my pluses. We're going to get the 100 microliters. Let's get that mixed up good. Okay. And 100 microliters. And 100 microliters. Okay. Okay, and then spread that out so any colonies will be nice and distanced from each other, socially distanced colonies. I can remove this. It's not a big deal because it's the same one. I'm going to stack these up and I'm going to put them in the incubator for 24 or so hours at 37 degrees Celsius, which is 98.6 degrees Fahrenheit. And then while I am here, I am going to go ahead and show you some plates from yesterday. So I can just go ahead and record this all at once. On my find my pen. Pen light. Pause this. Okay, so let's take a look at what we see under these plates right now without any sort of light. So this is the plate that did not get pGLO, and it's just the LB with E. coli, and you can see there is a ton of growth. You can't even see isolated colonies. It's just a ton of growth on that plate, okay? It's because it didn't have the ampicillin resistance gene because it didn't get there's no ampicillin here. No, so there's no ampicillin here. And so that's why we get a lot of growth. Next plate is our minus pGLO plus E. coli plus ampicillin. And I don't think there's a single colony on here, which is good because that shows us that we don't have any ampicillin resistance contamination here. And that worked just the way we predicted it. All right, this one is the arabinose. So this is the one we're expecting to glow in the dark. And the good thing is we don't have a ton of colonies here, but there's 4, and hopefully they will glow in the dark. And then this one, like I said, they should have more or less the same amount of colonies. And this kind of worked out perfectly. There's only like 6 colonies on here. But since this does not have arabinose, we're not expecting this one to glow in the dark. But it did grow relatively about the same amount of colonies. And it's only a few. I actually just upped the amount of plasmid for today to hopefully we get more. But it only takes one. If we get one that glows in the dark, then we know we were successful. So let me hit the lights. Get it nice and dark in here. And you can see nothing glows in the dark, but this is not a protein that glows in the dark on its own. It needs to have UV light shined on it. So this is the one that's plus pGLO plus amp, but no arabinose. So we're not expecting this one to glow. And let's see if we can get a good look at that here. Okay. Right. So that they're not glowing in the dark. You can't really see that. Like that's a better look at it like that. They're not glowing in the dark. They're just there. Okay. Let's take a look at these. And unfortunately, I could see them glowing in the dark really nicely here. I wish I can get this to look better on camera. They're bright green. Let me see the best way to show this. It's not dealing well with the light. That's a bummer. How it comes up on the camera. In person, it's quite dramatically green. Let me see if I can get this to be any different. See these? Do these look any different? Yeah. It's a bummer because in person it's like, I wonder if I can get it with my phone. In person, they're definitely green. Let me see if I can get this with my phone. Oh yeah, there we go. That's nice and much better. So I am going to take a picture of this and then show it with my phone. Oh, nice. Okay. So hopefully you can see this. Oh, that's just bad lighting. It's like overkill on my lighting. Let's see here. Bummer. Well, I'll post a picture too. There, that's a good look at it. That looks good. You can see in the dark colonies. Okay, and that's that.